All right, everybody, this is take number 2. On this episode of AP Biology Online, you're going to go ahead and learn about cellular respiration. So, cell respiration is a relatively long and lengthy topic. There are 3, kind of we can argue 4, maybe even 5 specific parts to it. So, we're going to get right into it. Giving you a heads up, there is a lot of information that you're going to need to be responsible for and being able to understand. So you may want to pause the video and just take a look at the specific requirements. I'm going to kind of give you the clip notes of it. Um, Basically, the first little bit of information is that everything ends up needing energy. So what is the method in which we do it? We do either fermentation, which is also called anaerobic respiration, or do cellular respiration, which is called aerobic respiration. Basically, these two different forms of energy production are how you're going to go get the energy. While fermentation is way less efficient, it is a way to go and generate your own energy in the event that you cannot do cellular respiration fast enough or you don't have mitochondria. The whole process is using enzymes. These enzymes will catalyze reactions, and we're going to go ahead and transfer this biological energy from carbohydrate sugars into ATP, adenosine triphosphate, as the end goal. There's just a bunch of different energy transfers that take place. Specifically, we'll be looking at these guys, NADHs and FADH2s. Bottom line up front, they are going to end up being reduced, meaning that they are going to go and collect an electron that they're going to go and deliver to the electron transport chain to power it up in order to do active transport. And basically what's going to happen is that that's going to go and generate all the ATP that we need. It is an aerobic process. You also have that there are aerobic prokaryotes that are able to do this as well. We're going to briefly mention that there is some prokaryotes that have an electron transport chain, which is found within the mitochondria. The reason why we talk about this is back in Unit 2, we talked about the endosymbiotic theory. The whole thing about that was that the one cell ate the other cell and then incorporated part of it. So we're going to go kind of quickly touch on bacteria basically having a, a the workings of an electron transport chain, but not actually in a mitochondria. But you can kind of use the connections of saying that, well, this is actually the bacteria that was the mitochondria, or at least evolved into the mitochondria. There's going to be just information about these different reactions. Oxidation, phosphorylation is going to be the one that's going to be used in order to go and create all of our ATP. And then we have these pathways right here. So we start with glycolysis. What happens to glycolysis? What's this pyruvate? Talked about the Krebs cycle, also known as the citric acid cycle, and what we're making in there. And then finally, the electron transport chain, oxidation, phosphorylation, chemoosmosis. This is all this words that I'm just kind of throwing at you that happens at the last step. And that's all the information that's in there. Okay. What you need to be aware of, right? Everything needs energy. Everything needs energy in order to live. Once you stop using energy, typically that means that you're dead, or you're dying if you're not using the energy effectively. So the input for energy, right, is going to be glucose. And what I'm trying to do is the end state of getting it into ATP effectively. Now, the two different ways that this works, right, we're going to be using this process that we'll call respiration, which is just basically energy production, and where it's going to be with oxygen, which is going to be aerobic, and then you have without oxygen, which is fermentation or anaerobic. But both cases, we're going to end up getting this ATP end state that's in there. In order to go and do that, we need to basically turn the glucose into ATP. I'm going to be using these electron carriers in order to go and do that this whole entire time, because electrons have a lot of energy associated with it. There's going to be all these neat chemical reactions that will take place, and we're going to go and show it off. All right, this is basically a picture that you'll find within your textbook if you've been reading it. And basically all this is showing you the process on how we start over here with glucose and eventually end over here to get our ATP synthesis at the end. ATP synthesis, I should say. the whole process ends up being a very, very long and lengthy process. We have glycolysis, then we end up going to pyruvic oxidation, your citric acid cycle, and then finally electron transport chain. The reason why I bring this picture up is that you can see that we start over here with glucose, but it's kind of an interesting thing for you to be aware of is that we are actually able to use more than just glucose in order to go get energy from, right? You have body fat, right? They're stored as triglycerides within your fat cells. Well, I can end up having a process taking place where I'm going to go ahead and burn these fat cells. I think it's called lipo hydrolysis. I think it ends up being. It's basically you're taking the triglycerides that we learned back in biochemistry and you're just breaking them apart and you're going to go get the energy associated with it, which makes sense because we mentioned about how these lipids are used for long-term energy storage, right? So there are pathways that we're able to go do and to be able to break down the lipids and turn it into, energy sources for us to go and use. Alternatively, there's also lipogenesis where if you have an excessive amount of glucose within your body and, we're not going to go store it as glycogen, well, what we're going to go then do is store it as body fat. And this is what happens when you consume too much sugary stuff and, we start to gain more body fat, which is just turning into our fat cells getting bigger and bigger and bigger. You're not getting more fat, you're just having the fat cells getting bigger to store more of this energy that's in there. So a little bit of a health background within it. Now, we're going to keep on going back into this. I would recommend that you go and grab this picture, or you want to go ahead and copy this like flow chart down here, and I'll go ahead and draw some stuff over here with you. Okay, but in short, when it comes to here, right, we have our cytoplasm right here, the mitochondrial matrix, and the inner mitochondrial membrane, right? So let's say that this is my cell right here, and then here is my glucose molecule, right? And then I'm going to go and, there's plenty more stuff going on in here, but just work with me, right? Here's the mitochondria, right? It has like this like squiggly looking shape that's in there. Keep in mind, right, the mitochondria has all these folds in here in order to increase the surface area, right, to volume ratio, going back to cells, right? It has all these folds in order to go and promote all of this ATP synthesis that's in there. So I try to make these connections as much as possible, right? So we have that. So glycolysis is going to go ahead and happen out of here. So you're going to have glycolysis take place out here in the cytoplasm, right? And then it's going to be modified into pyruvate. And then once it goes into there, then we'll have the citric acid cycle, or the Krebs cycle is going to happen within the mitochondrial matrix. And then the electron transport chain, can't really like draw it, right? The ETC is all abbreviated. It happens within this inner membrane right here where all the folds are, right? Where we're going to go try to generate that ATP. When we're talking about the inner mitochondrial space, the inner mitochondrial space, if I can just kind of change my color for a little bit, is referring to this area over here. So, and that's in between the two membranes. And this is where I'm going to be building up my proton gradient in order to harness some power, harness pressure in order to go and create my ATP. So what we're going to go do is basically go through each of these steps where it's like 1, 2, which is like a star because it's like kind of like an in-between step, 3, which is a big step, and then 4, which is like your last big step that's in there. Pyruvic oxidation is usually lumped in to either part of the citric acid cycle or glycolysis normally. I think most of the time we see it as part of the citric acid cycle, but we need to kind of recognize it for what it is and that it's its own little tiny in-between step. Now, really big thing to make you aware of is that you're not going to be responsible to know all the little tiny enzymes and all the in-between like molecules and stuff. We're just going to go and kind of get you an overview of cellular respiration. All right, and again, this is cell respiration. We are talking about the with oxygen reaction. Okay, so we're not doing fermentation. We're just doing cell respiration. Now, a little caveat to that is that glycolysis is going to be the first step to both cell respiration, and fermentation. Because you gotta kind of think about this, right? Like not every single cell, right? Not bacteria or cells, they don't have a mitochondria. So since glycolysis happens within the cytoplasm, right? Since this happens within the cytoplasm, everybody is going to go ahead and do this first step. So no matter what. So that's that. Now, the way that if we had to go and give you a too long didn't read, what is the whole purpose of glycolysis? We're going to go and break down glucose and we're going to go make some ATP, NADH, and pyruvate. Like, if anything, that's what you need to kind of get out of this. Like that is the main shtick and where it ends up happening. What I'm going to go do now is go into a little bit more detail about like how glycolysis works, just so that way you have a little bit of a background understanding of what it is that you're looking at. All right. Now, whenever I talk about glycolysis, I always mention it that it is a two-step process. Now, you're probably looking at, wait, is there a step 1, step 2, step 3, 4, and 5? Like, what do you mean two-step process? You don't need to worry about all these in-between steps. You don't need to worry about the names of the stuff that's in there. The only thing you really need to worry about is glucose and pyruvate, okay? Don't worry about this fructose 1,6-biphosphate. We'll explain what that is in a bit, though. When I say like two-step process, it's an input and output. Or another way to look at it is that it's an investment and then a payoff, right? Think about when it comes down to businesses, right? Why do you invest in businesses? You go watch Shark Tank, and they're like, "I'm going to go give you $1 million for 10% stake of your thing," and just, that whole thing, right? Well, why are they investing? Because they see the potential and they see the payoff, right? They know that if they put money into this business, they're going to go make more money at the end of it, and this is basically what ends up happening, right? In the case of glycolysis, we make 2 ATPs, I should say net ATPs. We make 2 NADHs, and we make 2 pyruvates. Very, very easy, 2, 2, 2. Now, if we go take a look, let's go take a look at step 1 and step 2. Notice that I have my ATP here, but its ATP is becoming ADP. So I'm actually spending 2. So in this first step, right, I spend 2 ATPs. Okay, so I'm actually just lost some ATP right now. That's not really ideal. Again, I'm trying to make energy, right? Well, keep in mind, right? I just invested. I put a little bit of input. I've just invested my 2 ATP. So what ends up happening is that I'm basically dropping off the phosphates, right? Because ATP is adenosine triphosphate. So one of the phosphates, right? Well, the phosphate from this ATP is being lobbed onto here and being lobbed onto there. And basically what I've done is I've now put a little bit more energy into this to be able to be manipulated. This fructose 1,6-biphosphate, which just basically means where is it that the phosphates are on there instead of the 1 in the 6 position. That's where the name comes from. And it's a fructose sugar, right? What's going to end up happening here is that we're going to go and split it in half. So every single thing that happens basically happens twice. That's why you see this splitting into 2 different pathways, but it's literally the exact same reaction happening twice. So we can see that we're going to go and manipulate this and to eventually get us to the pyruvate. So what I'll do is I'll just go ahead and show you on the left side and just, it mirrors exactly on the right side, right? So once I'm at here, after I've invested my 2 ATPs, I'm now going to go and generate some NADH. So how this reaction goes is that the NAD plus, let me go and get this right, NAD plus, right? It's very similar to NADP plus, right? This is the one that we found in photosynthesis, okay? And what these end up doing is that they're high energy electron carriers. And the goal is that these guys are going to eventually go to the electron transport chain to deliver their energy. All right, so I'm trying to make this specifically it's NADH. Basically, the NAD plus gained an electron, gained a hydrogen proton, and I'm going to go ahead and deliver this to the electron transport chain. So I'm really just trying to make these guys right here. Making ATP on top of it is kind of like nice, but this is where I can get the most bang for my buck. So I made 1 NADH, but being getting it right, there's 2 of these molecules I made, so this happens twice. So 1, 2, there's my 2 NADHs that I just accounted for, right? And then if I go take a look at the left side, look, I am now making ATP again. I made 1 2 ATPs. So I made 2 ATPs on this side, and I make 2 ATPs on that side. So I've actually made a total of 4, right? So I made a total of 4, but keep in mind is that I spent 2 of them, right, to do this. So really, I'm only actually netting 2 because I used 2 earlier. So this is what I'm saying. I net 2 ATP. That's the netting the 2. And then at the end, I make my 2 molecules of pyruvate. These 2 molecules of pyruvate are going to go ahead and be used to be modified in order to go into the citric acid cycle. Now, in the case of fermentation, we're basically going to take this NADH and just modify it a little bit. But right now, we have a final destination. We have a location that I can shepherd this NADH to in order to get more energy out of it. And that's going to be the next step. All right. What I've always found that's been helpful about this is just kind of doing a summary and kind of keep on doing the summary over and over and over again. Okay, so in glycolysis, we have my ATP, I got my NADH, and I have got my, let's say like CO2, because I want to keep track of this guy as well, and then my, what is it? Water as well. I can do like other products as well, but just kind of keep in mind, right? So far I've made 2 ATP, I made 2 NADHs, okay? One thing that I realized that's not on here that we need to go do is I need you to write down the cell respiration equation. And the equation goes like this. You have 6 O2, 6 oxygen, plus C6H12O6, 1 glucose, will yield you 6 CO2, 6 carbon dioxides, and 6 H2O, 6 waters. Okay, so if we go take a look so far, all I've done is that I've used the glucose. So of my equation, I have accounted for the glucose. So that's it. I've not used any oxygen yet. I've not created any carbon dioxide or water. And we're going to keep on going back to this equation over and over and over and over and over again. Okay, so of the stuff so far, I have made I've made 2 ATPs. I made 2 NADHs. All right, so we just completed this step. I'm now going to be moving on to the pyruvate oxidation. Pyruvic oxidation is a very, very, very simple in-between step that we don't need to, spend too much time in the weeds on. Okay, what am I looking at here? Basically, the pyruvate, right? Now, keep in mind, this is just one pyruvate, and how many do we make at the end of glycolysis? We made 2 of them, right? So you have to keep in mind that whenever you see anything like kind of going forward with the Krebs cycle, with anything that has to do with this pyruvate, it happens twice. All right, it happens twice. All right, so just kind of keep that in mind. All right, so I take this pyruvate. I'm going to go ahead and turn another NAD plus into NADH. So I've made some more NADH, and take a look. Now I have made some carbon dioxide. So if you think, right now you're breathing, you are taking in oxygen, you're releasing carbon dioxide. This is where all the carbon dioxide comes from. This is where all the carbon dioxide that comes from in your body. All your, 300 trillion cells are doing this cellular respiration, and they are generating carbon dioxide, and you are releasing it, right? So it's kind of like neat that this is where it's coming from. So I've made NADH, I've made carbon dioxide, and then I'm also going to make this acetyl-CoA. This acetyl-CoA is just an in-between step to allow it to go into it's able to go into the Krebs, into the citric acid cycle. Oh, citric acid cycle. Don't need to put an A there. All right, now keep in mind, right? This happens twice. So I've actually made 2 NADHs so far. I've made 2 carbon dioxides. I've made 2 acetyl CoAs. Now, what I want to do is I'm going to keep moving forward because it's going to make a little bit more sense to go and move forward. Okay. Now, the whole thing is that we are going to be doing some oxidation reduction scenarios, right? This guy ended up being reduced, so this one ended up becoming oxidized, and it's oxidized. Now I'm able to go and have this molecule here in order to go forward into the Krebs cycle. Phase two, right? So again, like the last thing that was kind of like an in-between step, right? Here's that acetyl CoA that we just made, right? So again, this is going to happen twice. And I keep on bringing this up over and over and over and over and over again because I want to make sure that you know that this is happening 2 times. Do you need to know all the steps of the citric acid cycle, the Krebs cycle? No, you do not need to know about all the different steps that are in here. It's ridiculous. You don't need to know them, okay? I'm going to ask, what are we doing? We're going to be releasing more carbon dioxide. We're going to be making some ATP, and I'm going to go ahead and make some NADH and FADH2. When it comes to my high energy electron carriers, NADH is one of them that helps out in cellular respiration. FADH2 is another one. How's it different? Don't need to worry about the weeds of that. FADH2 just affects another part of the electron transport chain, but for bottom line up front, it provides energy in the form of high energy electron carriers. And what happens? We take this acetyl CoA, and we're just going to go through this cycle right here, right? It gets modified, and then once we're at this step right here, I'll throw another acetyl CoA, and I go through the process all over again. All right, let's go take a look at the products that I'm making during this. And then that way you should have a bit of a clearer idea of which it is that you're looking at, right? So let's go and do a little bit of a count, right? So I got NADH, I got ATP, I got FADH2, and I got carbon dioxide. These are the 4 things I'm making during this, right? So if I count, let's go ahead and count my NADHs. So I got 1, 2, 3 NADHs, right? So I have made 3 NADHs, but keep in mind, this happens twice because that there are 2 acetyl-CoAs, because there are 2 pyruvates, because I make 2 pyruvates with 1 glucose. So I'm actually making 6 NADHs total, right? So the first number is going to be per pyruvate. This is going to be per glucose. Okay. Changing up the color, let's go ahead and do another one. All right, so let's go ahead and now do my ATP. Now this is weird. You'll see that we have this GDP plus phosphate group equals GTP. There are times that people will write down ATP, GTP. This is basically the ATP, right? So we made 1 energy right there, but keep in mind, this happens twice. So there is actually 2 of these phosphate holding molecules existing in here. Next, we'll go talk about our FADH2. We go take a look around. I make 1 FADH2. So I make 1 for a total of 2 because there's going to be 2 turns with the cycle. And then finally, there's going to be this carbon dioxide. And I make 1 carbon dioxide here. I make 2 carbon dioxides right there. So I made 2 for a total of 4. All right. Now, one thing that I would like to go do is I'm going to go back a step, and I want to just talk through the in-between step of us taking the pyruvate and turning it into acetyl-CoA. If you remember, what else did we make right there? We made 1 more carbon dioxide per pyruvate, so 2 per glucose, right? And then we also made some NADH. And for my NADH, I made 1 NADH, which meant I actually made 2 of them right there. All right, so why am I bringing this up? I'm bringing this up because we need to account for everything we've made so far. So in total, per glucose, I've made 8 NADHs, I've made 2 FADH2s, I've made 6 carbon dioxides, and I made 2 ATPs. So what I want to go do is I'm going to go backwards, right, to that, the chart, and let's go and see if we can kind of keep track of everybody, right? So we're here. Right, so if I go back to ATP, I go back to NADH, I go back to CO2, to water, it's just like H2O, and also I want to do FADH2 now, right? Let's keep in mind, right? I made 2 ATPs and I made 2 NADHs with glycolysis. And pyruvic oxidation and the citric acid cycle, I'm combining these guys together. I've made 2 ATPs, I've made 8 NADHs, I've made my 6 carbon dioxides, I've made no waters, and I made 2 FADH2s. So if I'm totaling all this now, I now have 10 NADHs, 8 and 2 is 10. So I now have 10, right? So I'll do like the total here, right? I got 4 ATPs, I got 10 NADHs, 6 carbon dioxides, and 2 FADH2s. If you go back to the equation, right, we did the O2, 6O2 plus C6H12O6 yields you 6H2O plus 6CO2, right? I've now have my CO2 accounted for because I made it in the citric acid cycle. Glucose is accounted for because I used it back in glycolysis. So the last step is that I'm basically going to be taking this oxygen. This is the oxygen that you're breathing in. This is why you need oxygen. This oxygen that you're breathing in is eventually going to become water. And it's kind of like crazy to think about, right? Here's actually where you can like see that like happening right in there. Specifically, it's the water right there, right? Because we've already made the carbon dioxide. The ATP goes off, bumbles around, is going to go and get used as high energy. Your adenosine triphosphate used as an energy resource. But now this NADH and this FADH2, we're going to go use these at the electron transport chain in order to go and power it up. And this is where the craziness starts to happen here, okay? NADH and FADH2 are going to transfer those high energy electrons that they've accumulated from glycolysis in the Krebs cycle to the electron transport chain, and that's going to be where you get all your energy from. This is in basics, what's actually happening, what we're going to go and see, okay? There's two parts to the electron transport chain. You have these carrier proteins that are shepherding hydrogen ions from one side to the other. These hydrogen ions come from the following place, right? So the NADH, right, and also the FADH2, they're going to basically become, right, the NAD plus your FAD as well. And we're breaking these down into the high energy electrons, which are going to be used in order to go and power these proteins right here. Think of it like as electricity, really just like passing this electron around that's going to go and change the shape of these proteins, which is going to allow them to go and do work. We're also, when you break apart this high energy electron, you're also making all these hydrogen ions. This is where the H pluses are coming from. So all these H pluses that you're seeing all over the place are coming from these guys right here. It's kind of neat, all right? And then we're going to go explain where these end up going at the end because they have to go somewhere, right? And then the NAD plus and the FAD, this is going to go back to the cytoplasm or the mitochondrial matrix in order to become more NADH and FADH2 again, right? So once I end up using it, it's going to go bumble off, go where it needs to go, and be used once again. So the high energy electron carriers are going to go and drop off their electrons, power up these pumps. All these hydrogen ions that have been accumulating from them dropping off their electrons are going to go pumped into this inner space, mitochondrial space, which is in between, right? If we go take a look at the mitochondria again, right? We're pumping it into this spot right here. So this is the inner membrane. This is the inner membrane, which is like right here. And then you have to use your imagination that the outer membrane would be like located, further up in there, right? So there's only so much space for these hydrogen ions to hang out in. And what's basically going to happen is they're going to go build up pressure. We're going to go harness that pressure, push them through this enzyme, and we make ATP at the end of it. That's basically what ends up happening, right? So we power some proteins, we pump some hydrogen ions over here, we build pressure, we harness that pressure, and we make energy from it. Right? If you need to, like, just re-watch that, but I'm going to go ahead and move forward just for the interest of time. This is the electron transport chain. It should look almost identical to the one that you saw back in photosynthesis, right, in the little handout that I gave you. I don't go into a lot of detail about it because it's relatively the exact same process. Like fundamentally, it's the same process. But the difference here is just, I use NADH and FADH2 instead of NADPH in photosynthesis. And look, everything I just talked to you about, right? Here's my NADH, there's my FADH2. What do they do? They're dropping off their electrons. They're dropping off their electrons at these two locations. They become NAD plus and FAD. That's going to go back to the mitochondria matrix, or it's going to go to the my thing died for a second. Okay, there it goes. Where was this thing? Okay, so the NAD plus and the FAD is going to go back to either the cytoplasm or the mitochondrial matrix in order to go and be turned into NADH and FADH2 again from either one of those reactions. I drop off my electrons. The electrons basically work like electricity, and they're going to go and power up these specific proteins right here. What are the names of them? You don't need to worry about it. It's fine. And what happens when it passes through the hydrogen ions that were attached to the NADH and the FADH2 are going to be pumped into this space, this inner mitochondrial space. And there's the outer membrane that we talked about, which is the outer membrane, the outer portion of the mitochondria, and then the inner membrane where all the different folds are right there. So I am pumping all these hydrogen ions and I'm building up a proton gradient, right? And this is actually a form of active transport, giving you a heads up, right? Because keep in mind, I'm pumping hydrogen ions where they don't want to be because I'm going against the concentration gradient. These guys want to go this way or that way, but they can't because the hydrogen ions cannot diffuse past the cell membrane that's right here, right? This is a membrane. It's a phospholipid bilayer. We know that's hydrophilic heads and hydrophobic tails. These hydrogen ions don't want to be anywhere near those hydrophobic tails, so they can't get out. And what I do is I build up pressure, like as if I'm blowing up a balloon. And what I'm going to go do is I build up so much pressure, the balloon gets so big. I'm going to go and just let a little bit of air escape it at a time. And every single time the air escapes it, I'm able to go and generate an ATP molecule. And it is great on how this ends up working. All right. Now, the last bit that I want to draw your attention to, I'm going to go ahead and kind of reset this, is look over here. Here's our oxygen, here's our water. Okay, so oxygen is O2, right? So if I split, and then your H2O, right? If I go take a look, I got two oxygen atoms. I got one oxygen here, and I got two hydrogens. Well, the oxygen is becoming oxygen. That's perfectly fine. But where are the hydrogens coming from? Let's take a look. The hydrogens that are being pooped out right here from the ATP synthase are going to go ahead and be collected by this oxygen in order to become the water. But now, right, hydrogen ions, H pluses, they need an electron in order to go ahead and be stable. Where am I going to go get an electron? Boom. This electron is going to end up being where this oxygen is. And the oxygen is going to nab that electron. It's going to nab that hydrogen ion, the hydrogen proton, and then it's going to just do some chemical reactions and it becomes water. So water, or I should say oxygen, is the final molecule in cellular respiration that gets reduced, right? It's going to be reduced. I'm going to go and collect that electron. I'm going to go and collect my hydrogen ion. I become water, and that water ends up being diffused out, goes somewhere, right, and ends up, going off wherever. And gang, that's pretty much what ends up happening here, right? So the NADHs and the FADH2s, we're going to go pump all this energy. So they're going to pump all these electrons in order to go and build that gradient. And then eventually we're going to go kick it out and create all the ATP that we need in order to go and keep the cell functioning. This is just an image to show you where an electron transport chain can be found within bacteria. And I'm not going to go and bore you with the details. You can pause the picture and take a look. But basically what it ends up doing is that it shows you that the similar reaction is going to be happening on the membrane of a bacteria. They have a couple little weird things that are going on the outside of their cell membrane. It's not anything you need to really be aware of. But basically the idea is that we can find the schematics, a very, very primitive electron transport chain on their cell membrane. And what we can do is come up with a hypothesis that this supports the endosymbiotic theory, that one cell ate a bacteria cell that had this electron transport chain that was more effective at making ATP. And instead of breaking it down, it was better for me to just hold on to it. This goes into detail about what I was mentioning earlier. The NADH and the FADH2 will pass their electrons off, and the proteins will just hold on to them, pass the electron forward, and then during that time, we're going to be pumping those hydrogen ions, right? Again, photosynthesis, it's NADP+. my way I always kind of remembered it is that NADP, P for photosynthesis. If there is a P present, it's the photosynthesis high energy electron carrier. NADH, FADH2, they do not have a P that's not for photosynthesis. Just another picture that ends up showing you the process on how this ends up working, right? I basically walked you through the whole entire process on the first page, but it's just you're building a proton gradient, you're building up pressure, you're going to end up harnessing that pressure in order to go and create your ATP. They go from areas of high pressure to low pressure. That's providing the chemical potential energy to turn that into kinetic energy. Sorry, I should let me take a step back. There's potential energy in the mitochondrial space. When the potential energy goes through ATP synthase, it turns into kinetic energy, and then that kinetic energy will be harnessed to create chemical potential energy in the form of ATP. This is where we end up talking about oxidation phosphorylation, where we are creating our ATP. And photosynthesis is called photophosphorylation. So phosphorylation, just you're adding your phosphate groups on there. It's all these coupling reactions where you have these oxidation reduction reactions, and the electron transport chain is just there in order to go and help facilitate these reactions. It's going to be generating heat. We are going to be releasing a lot of energy, so we're trying to be as efficient as possible. This ends up giving you that one-stop shop of everything that happened, and all the the process of creating our ATP and the different steps that are right there. Right. Keep in mind, if you don't have a mitochondria here, you are still able to generate ATP in the form of glycolysis, but the mitochondria is a little bit more effective. The actual math behind this kind of argues from person to person, but here's the kind of way to look at it. You will get ATP from glycolysis and citric acid, right? You get 2 and you get 2. So we at least napped 4 right now. NADH has been argued that it can make 3 ATP per NADH molecule, and then FADH2 can make 2 ATP per molecule. So if you remember, we made 2 NADHs in glycolysis and 8 within the citric acid cycle and the pyruvate oxidation. So 8 times 3, you're going to end up getting, 10 times 3, you'll end up getting 30 right there. And then FADH2 has been argued to make like about 2 ATP each. So you have 2 of them that were made in the citric acid cycle. So that's 4 ATP and then 4 right here. So, doing that math, it's 38 ATP. But you're going to go find that the literature they will include certain steps or not include certain steps. So you'll usually see the literature sometimes is like 36 to 40, depending on the stuff that's in there. I'm not going to go and ask you this. It's not really super relevant to get into the weeds, but I guess what I want you to be aware of, right? Glycolysis is only making 2 ATP, while in the case here, we're making like 36 within the mitochondria. So having a mitochondria, right, when we say that it's the powerhouse of the cell, and I hate that because it's such an oversimplification. Nah, it's a site of excessive ATP synthesis. And this is that ATP that is going to be used in order to go and drive those chemical reactions and allow your cells to be able to do all the really cool stuff that's in there. We'll end up going into fermentation, another class. Basically, it's that you cannot move forward past glycolysis. So how is it that we end up moving forward? We'll end up showing you it. We'll show you lactic acid fermentation and alcohol fermentation, and how that ends up working when you get back from break. And then this is just showing you the coupling reaction and how like ATP and water ends up becoming a adenosine diphosphate, just how this ends up going. So you're going to use water in order to go and lop it off and be able to harness your energy. That energy gets used in order to go and make chemical reactions take place. And then at a certain point, you got to repower that ADP, which you do at an ATP synthase. Or I guess in glycolysis, where you go and make your ATPs there and in the citric cycle. All right, hopefully the audio worked on this one. And yep, come in with your questions, and I will catch you on the next one. See you guys.